Is now starting. All attendees are in listen-only mode.
Hello everyone, welcome to Queensland Water's Essential webinar. I think this is our third one. It's been a really good series so far. We've had a lot of positive feedback. So before we just get started, you'll notice that I'm running the webcam and you'll have Rob there as well. We've had some attendee feedback that you wish to see our pretty faces for the start of these, to see who was talking. Who these lovely nasally voices belong to. So just before I hand over to Rob to give that COVID industry update and talk about our lovely presenters today, just some housekeeping things. So regarding the questions, if you've been a part of one of these webinars before, you'll know that you can raise a question through the question box, which is usually on the, it's all backwards for me, on the right-hand side there. Or you can raise your hand, which is on that little grey panel. I have lovely colleagues messaging me and trying to make me laugh on screen. So questions, raise your hand if you wish to actually throw it out to the group and with your lovely voice there. And finally, regarding the webinars, we've had people that can't attend these. So we're looking at putting these webinars online so you can go back and watch these later on, or just access the slides and the questions and answers that are relevant to each presentation. This, of course, will be, we'll check with the presenters that this is okay, seeing as sometimes there's a bit of, items within the presentation that may not be publicly released. So I'll hand across to Rob now so he can give a bit of an update.
Hi all, got two great presentations today, so I won't take too much of your time. I just wanted to mention that the Zoho forum's been updated, and yesterday I sent out a message to the whole group talking about the resources page. So we've collected the links and information sheets that we've been sharing for the last few weeks all into that one page, and I was asking for feedback on whether not whether that's a valuable resource, that'll stay there regardless, but whether you want to see that updated. I'm just a bit nervous that people are being overloaded with information now. We started with a lack, but now we're probably getting too much, and there's plenty of good stuff floating around. We'll keep sending out messages, but if you want to provide feedback on the best way for us to share links and resources with you, be keen to hear it. Just email me or give a thumbs up on that resources page if you're on the Zoho forum. If you're not in the Zoho forum, send me an email and we'll hook you up. There's some recent news there. So yesterday, the Department of Environment and Science released their approach to regulation during COVID duress. And we were talking yesterday to the excuse me, to the water quality regulator who have decided on their approach now, and we'll be emailing that around today as well. So let's get on with the presentations. First up, we have Professor Stuart from University of New South Wales. He's doing a general talk. We called it COVID Facts in the Water Sector, but many of you will have seen Stuart's blog that's on the resources page, where he covers all things science and COVID, I guess. I'll hand over straight to you, Stuart. Thanks for joining us today.
Thank you. Thanks, Rob and Ryan, for the invitation today. Hopefully everybody can see my screen. So yeah, I'm going to go through a basic rundown of what we do know and what we don't know in terms of the virus that causes the illness, COVID-19, around water and water management. So the virus itself, as most people would know, has now been formally named SARS-CoV-2. So the SARS coronavirus #2, indicating that it is a very close relative of the original SARS virus, which was referred to as SARS-CoV. So most people would have seen plenty of diagrams by now, I think, in describing the physical characteristics of the virion, the virus particle itself. But there are a couple that are really important for understanding how the virus behaves in water and what the risks associated with it may be. So I'll just quickly run through. Right inside in the virus is the genetic material. So this particular virus is an RNA virus, and we'll come back to that point because it has implications. So it's a single-stranded RNA virus. That RNA is coded in a protein called a nucleocapsid. So the nucleocapsid protein, which is what's in here. And there are three other kinds of proteins that the RNA codes for, which are referred to as envelope proteins, membrane proteins, and this spike protein, which is responsible for latching onto the cell receptor and invading human cells. And the other key component of this virus, as people would have heard, is called the lipid membrane. So there's a lipid membrane made out of lipids that surrounds the virus, which is a relatively unusual feature as far as many other viruses go. And it's the reason that this virus is particularly susceptible to soaps. And so one of the reasons that people are suggesting that we use soap to wash hands, etc., because this lipid membrane here is quite unstable when it comes into contact with soap. And this entire structure is required for the virus to be a viable infectious entity. So therefore, any part of this virus that we can start to attack will reduce the viability and reduce the survival of the virus. So there are proteins, there's genetic material, RNA, and then there's the lipid material as well. So some of the things that we know about enveloped viruses, so having that lipid envelope around them, they can survive in water, and we'll look at some data to support that. But all of the research shows that they tend to be much less persistent in water compared to the non-enveloped viruses. So that characteristic in itself is important. And all of the viruses that you would be familiar with as waterborne disease concerns, a number of them listed here, they're all non-enveloped viruses. So I think there really is an important distinction there between being an envelope virus and a non-envelope virus, and being a major waterborne public health concern or not. If we look at the physical size of the virus, this diagram was produced prior to SARS-CoV-2, but as we described, it's very similar to the SARS virus itself, SARS-CoV-1 we now call it. And in terms of the size changes a little bit depending on whether we're in a salty saline media or a clean water medium, but around about 120 nanometers is the particle size. So if we're thinking about water treatment processes that are size dependent, reverse osmosis, filtration, etc., it's certainly a lot bigger than some of the others that we're very familiar with. Poliovirus is a very simple virus, but also hepatitis A virus. These tend to be much smaller entities than the virus that we're talking about now. Do we excrete this virus in feces? So there's a lot of evidence now, both showing that we can measure the RNA of the virus in feces as well as in wastewater, and we'll come back to that. But that RNA is only one small component of the virus. It doesn't indicate that there's a viable infectious entity present. Whether or not infectious viable SARS-CoV-2 virus is excreted in feces is still a bit of an open question. So there have only been a couple of studies that have attempted to actually culture the virus, to actually grow the virus and show that it's present. And both of those are not particularly indicating that there is a lot of live virus present. One study out of China reported being able to culture 2 out of 4 samples, and they identified a live virus, or at least a full viral structure, using electron microscopy. So not necessarily infecting cells directly, but showing that the structure was present. Another study attempted to culture fecal samples and was unable to do so. But that's a pretty small amount of evidence, data. All viruses grow differently in different media. I think it's a reasonable suggestion to say that we've still got a long way to go in terms of understanding how best to culture these viruses and show whether or not they are viable in fecal material. And there are reasons why we are worried about that possibility. The main one coming from the SARS virus in 2003 in this particular incident or case study in Hong Kong, a large domestic building known as the Amoy Gardens. And in this circumstance in 2003, one of the flats here, so the way the building is arranged approximately looks like this. We've got flats 1, 2, 3, 4, 5, 6, 7, 8 going around the building. So one of them, of flat 7 on floor 16, there was one infected patient who responded with diarrhea and had extreme diarrhea apparently, which was then flushed down that person's toilet. This is the external plumbing on the building, as you can see. And when they looked at what happened in this building, all of these grey coloured flats all became infected. So on this side of the building, up and down through multiple floors, there was a lot of research. The World Health Organization was involved in having a look at what effectively happened here. And the conclusions were, this is the external plumbing on the Amoy Gardens building here. The conclusions were that you have all of these different drains coming out of the bathroom. You have the toilet, you have the hand basin, and you have the floor trap. And the floor trap was coming out through separate pipes, and I think all of them were coming out through separate pipes, joining onto a main wastewater pipe coming down the building. And each of them, some of them had a trap, a U bend underneath them. You can't necessarily see them in this pictures. This is another one. This is not the Amoy Gardens. It's another building in Hong Kong, but you get the idea here where you might have a floor trap coming out, a toilet, a hand basin, etc. There may well be a U trap for each of these, but they're on the individual pipes themselves rather than a common trap. So what had happened was that the floor trap had dried out because it hadn't been used, that nobody had mopped the floor for a while, and therefore there was effectively no trap. And so you have potentially aerosols and vapors being able to move up through this system into the floor trap, being drawn in through by the air conditioning in the system, which played a role as well. And therefore, that's how they believe all of those people became infected on all of those other floors. So since then, they've gone back and they've retrofitted a new system which effectively has a common trap. So you've got the floor drain, the bathtub, and the wash basin all coming down to a common trap so that it can't dry out simply because one of them hasn't been used. So that's one of the reasons that people are concerned about exposure and transmission via wastewater. So quite a bit of study came out of that SARS experience. This is one study that was published in 2005, a couple of years later, where they started to look at the persistence of the virus in wastewater. They found that at 20 degrees Celsius in hospital wastewater and domestic sewage and dechlorinated tap water, they found that in the fecal material itself, the virus could survive for about 3 days. 14 days in a phosphate buffered saline solution, so a saltwater solution, and about 17 days in urine. However, compared to this 2 days up here, they found that it actually lasts a lot longer in different samples, 14 days in wastewater, if it's cold at 4 degrees Celsius. And we've seen lots of studies now that are consistent with this. It tends to die off relatively quickly at warm temperatures, 20 degrees, but persists for a lot longer at cold temperatures, 4 degrees. Research has also shown that it is quite susceptible to chlorine disinfectants, more susceptible than E. coli, which is very convenient because E. coli is what we monitor and what we know we have lots of in wastewater. If we can continue to monitor good disinfection of E. coli, then that's a pretty good indicator of good disinfection of coronaviruses. And also colophages that have been used, that are used to do a lot of the research to understand disinfection chlorination. Free chlorine was more effective than chlorine dioxide, and some indications down here. More than 0.5 milligrams per litre of free chlorine in wastewater was shown to achieve complete inactivation down to the level of detection of their culturing methods. whereas more than 2 milligrams per litre was required for chlorine dioxide. But in both cases, more effective than E. coli and phage. Other research that's looked at the survivability of enveloped viruses in general. So this research looked at one particular coronavirus. This is a mouse hepatitis virus that infects mice. These are bacteriophages. People do research on surrogate viruses like these because they're not human pathogens, which reduces the level of containment that's required for the research laboratories to be able to undertake this work. And again, this has shown that at warm temperatures in wastewater, some of these viruses, some of these envelope viruses tend to survive for periods of time, 7, 13 hours for different viruses here, compared to the non-enveloped viruses, which survive a lot longer in those sorts of environments. Similarly in tap water, so this is dechlorinated filtered tap water. These are 2 coronaviruses here. One is a common human coronavirus, not the one that we're dealing with at the moment, and a feline infectious coronavirus was shown to observe numerous log reductions. So these are log reductions going down here over a period of 5 to 10 days, whereas poliovirus, which is a non-enveloped virus, was much more persistent in that water, and we didn't see the same degradation of the poliovirus. But again, it's temperature dependent. At 23 degrees, we saw the significant decrease of the enveloped viruses, or the coronaviruses in this case. At 4 degrees, it was much more persistent. And again, more research, again, with different surrogate viruses showing the same sorts of trends. These are all coronaviruses, or both coronaviruses in this case, and again showing that in reagent water, which is sterile laboratory grade water, the viruses were much more persistent than they were in lake water. So where you've got some natural organic matter present and whatever other factors might lead to faster degradation or inactivation of the viruses. And similarly, comparing 4 degrees with 25 degrees, at 4 degrees, some of these viruses showed almost no inactivation, whereas at 25 degrees, there was considerable inactivation over periods of days, talking about 22 to 17 days for about 2 log reduction in these cases. The genetic material of the viruses is both interesting and important for understanding persistence and for understanding inactivation, particularly with ultraviolet disinfection. So I did mention that this particular virus is a single-stranded RNA virus. So there are viruses that come as double-stranded DNA. There are viruses that have genetic material which is double-stranded RNA. And there are viruses of both single-stranded RNA and single-stranded DNA. As you know, RNA and DNA are composed of different nucleotides. These components here with nuclear bases on them bonded to the double helixes or single helixes of the sugar phosphate backbone. DNA has two different pyrimidine bases, thymine and cytosine, and 2 different purine bases, guanine and adenine. And RNA differs in this particular pyrimidine base, uracil instead of thymine. So why this is important, well, a couple of things. First, if we look at the RNA, the SARS-CoV-2 RNA, we know that it's about 30,000 nucleotides long. So about 30,000 of these individual bases up through the RNA complex. And we can find the genes for the key proteins within that RNA. So there's a gene that codes for the spike protein, S genes. There are genes that code for the E, the envelope proteins, the membrane proteins, and the nucleocapsid proteins that coat the RNA. So all of the important components and functions and other genes that produce enzymes and things that the RNA uses or the virus uses to replicate are all present, of course, in that RNA structure. And the particular genes obviously are minor components of the overall 30,000 nucleotides. Why that's important, and this is, I think, a really interesting paper that came out of Krista Vigenton's group at the University of Michigan, is she showed that if we use free chlorine to disinfect or inactivate the virus, free chlorine has effectively no impact on the lipid. So we've got the lipid membrane on the outside. The chlorine itself is able to penetrate the lipid membrane and makes its way into the inner parts of the virus. And what they did is they monitored the proteins. They looked at changes or oxidation of the protein during exposure to free chlorine, and they saw that the protein is the thing that degrades quickly. So the free chlorine attacks the proteins, and particularly the nucleocapsid proteins on the inside. We don't know why, particularly those proteins, but they're the ones that react the most quickly for the RNA viruses that they looked at, or for the envelope viruses that they looked at, and that pretty much dictates the loss of infectivity. So as the proteins are degraded, the infectivity comes down. So the proteins are like the weakest link when it comes to chlorine disinfection. When it comes to ultraviolet disinfection with UV254, we see a different effect. Again, we see no impact of the lipid. The lipid is not changed. It also doesn't provide any protection to the virus. The UV radiation penetrates the lipid membrane itself. We see no change really in the proteins. So the UV radiation has very little impact on the proteins. But what we do see is changes in the genome, the RNA. So it's the RNA itself, or the DNA for some viruses, that is inactivated by the ultraviolet disinfection, and that dictates the loss of infectivity. That's not a surprise. That's something that we know. So when we look at the germicidal portion of ultraviolet radiation, the UVC range up to about 280 nanometers, we know that there are a number of mechanisms. There are indirect photolysis mechanisms as well, but really the most important mechanism is the light itself inactivating genetic material, RNA and DNA. And what's actually going on there is that we have these base pairs that we spoke about, thiamines and cytosines, guanines or uracils in RNA, and it causes dimerization. So you might have RNA doesn't have thiamines, but when you might have two consecutive base pairs that thiamines, and you get this covalent bonding between them. So they bond together, and once you have a bond like that, it disrupts the nature of the DNA, and it's therefore unable to do all of the things that the DNA is supposed to do, and ultimately the virus becomes non-viable. And we can get dimers between all of the pyrimidine bases. So the TT dimers are the most commonly studied. They don't apply to RNA viruses, but we do know that we can get dimers between cytosine, and uracil bases as well. And so that explains why UV inactivation is effective for viruses. This group also did some really interesting studies where they looked at the naked nucleic acids. So they got RNA and DNA and exposed different types of RNA and DNA to UV254 on its own, and then they did it as the native virus particle as well to find out whether or not the virus structure, the envelope, etc., did provide any protection. They confirmed that it didn't. You get the same rate of inactivation regardless of whether it's naked RNA or whether it's part of the DNA structure. But they also showed that when they look at the rate constants, the degradation rate constants, they saw that single-stranded DNA degraded faster than single-stranded RNA, which was about the same as double-stranded DNA, which again was faster than double-stranded RNA. We're working in here. This is the coronavirus that we're looking at. So without any more information than knowing that it's really all about the DNA and the RNA, and the combinations of how you put together DNA and RNA are all about those four different bases. we can probably draw the conclusion that SARS-CoV-2 has roughly the same susceptibility to UV as other single-stranded RNA viruses. And if you look at the list of them, there's a lot of RNA viruses that we're familiar with and that we routinely worry about and do risk assessments about for drinking water and wastewater. Probably also roughly the same as the double-stranded DNA virus that we're familiar with, adenoviruses. And greater susceptibility to UV than the double-stranded RNA viruses, rotavirus. So that's all useful information. I won't dwell on this one too much because there's going to be a lot of discussion about it with some great work coming out of Queensland this week. So this particular paper that was published, but people showing that we can measure strands of RNA of SARS-CoV-2 in wastewater. There are a couple of different papers that are being produced now, one from the Netherlands, one from the US in collaboration with Singapore, and this one from Queensland through CSIRO and Joachim Mueller's group at UQ. And effectively, as it says here, a proof of concept that we can measure these RNA strands in wastewater. All of those papers, they're all looking at different genes, some of them looking at the nucleocapsid genes and the envelope genes, some of them looking at the spike protein genes. So there's a lot of standardization that needs to be done, but I think it's probably good that they've started off in diverse ways. Hopefully we'll work out which are the most effective ways for monitoring the virus in wastewater. So they're looking at different oligonucleotides. So these are the strands here that they're looking at. It's important to note that we talked about how this RNA is 30,000 base pairs or bases, nucleotides long. We look at sequences that are 20 or 30 nucleotides long. So there's really no indication that there's a viable virus here. It could just be scattered little pieces of RNA that are ripped apart and sitting around in the wastewater. So it's important not to draw that extrapolation that because we see these bits of RNA, therefore, we've got either a 30,000 long piece of genetic material or an even bigger extrapolation that there's a viable virus associated with that. Another couple of things to quickly mention, there is an interesting preprint that was produced about a week ago talking about what are we going to do when we go back to all of these buildings that have been shut down during COVID-19. And thinking about how safe or what sorts of water quality impacts we might see within particular water supply systems. It's an interesting paper, and I can give you the details are up here. You can probably search for it quickly, but I can send it to people if you're interested, showing that if we have no water use going on in a building, there are all sorts of water quality effects that we might expect to see around microbial growth, around etc., that might do water quality risks, and they go and they steps that people taking in to manage the risks of stagnant water that's been sitting in buildings for periods of weeks. And they talk
Stuart,
about how it's not just
jump in. We're just going to have to hurry things along, or we're going to run out of time.
Yep, good. So I am done. Just to say there are a couple of important fact sheets that are out there that people should be aware of. This one from the WHO and these ones from WAS Australia are definitely worth being familiar with. And this is the blog that Ryan mentioned, I think, at the start, or Rob mentioned at the start, that a lot of this information is being summarised on if people are interested to follow. That, and sorry I did go over time.
No, that was really interesting. Thank you, Stuart. We've got a couple of questions. I think we're going to have time for a lot. Those links that Stuart's put up there are all on the resources page already, and we will make this presentation available too. So if you can't get them, just give us a yell. Any questions, Ryan?
So yeah, as I said before, just submit your questions, guys. To get us started, Stuart, we've just had one regarding the temperature. So if temperature is important to the longevity, do you know if there's any evidence to suggest impacts are different in tropics versus temperate climates, or say in the different seasons?
And that's the obvious question, isn't it? I think that from bench scale testing in the lab, yes, it does appear to be a very consistent and reliable correlation that the virus survives cold temperatures much more than at longer temperatures. In terms of actual studies out in the field, or even epidemiological connections to that assumption, I don't think we've seen it. I haven't seen that question asked in a serious way. But I think it's a pretty good hypothesis that you're going to get much better survival in colder climates than you do in Queensland.
Excellent. Well, I'm sure some people are glad to hear that. So I've got another question from the audience, just in the interest of time. Compared to E. coli, how much UV intensity is required to kill COVID or CoV2? Yep.
So that particular question wasn't addressed in this paper compared to E. coli, but sorry, where was it? But what we did, what the researchers in this case did observe, is that it is all about the genome as I described it. So it's about the RNA or the DNA for the particular organism. And what they've shown is that the other components of both E. coli bacteria and of enveloped viruses have very little effect. There's very little protective effect of the cell membrane, for example, for bacteria. And there's also very little increased susceptibility because UV doesn't really react with in a strong way or in a rapid way with the components that build the rest of the cell or the envelope on an enveloped virus. So what it really comes down to is that they're all really similar, because it's the deciding factor is predominantly related to whether we're talking about double-stranded DNA, single-stranded RNA, etc. And even then, when you look at the data, there's not a big impact. So the UV disinfection is somewhat non-discriminative. It's reliable for all of these organisms because they all have exposed, susceptible genetic material.
Excellent, thank you. So we'll just wrap up there in the interest of time. Thank you again, Stuart. I'm sure the audience is silently applauding in the background. So those that submitted questions that we couldn't get to, I'm sure we'll be able to get Stuart to answer them offline, and we'll get them sent back out. So thank you again.
Pleasure, thank you.
You can imagine the applause in the background.
Yep.
Thanks very much, Stuart. We'll keep moving. We've got another great talk from Lynne Powell from Cairns Regional Council. She's going to talk about the development of their organic waste roadmap. It hasn't been complete yet. It's still under development, but I'm sure we'll hear how it's going. Everything's up in the air now, not just with this, but everywhere, I guess, with COVID. So I guess, Lynne, you'll be able to give us some idea of where things might head in the future. All right, so we'll hand over straight to Lynne.
Good morning, everyone. Thanks for having me for this presentation. It's not going to be as scientific or technically interesting as that last one. That was an absolutely brilliant presentation, thank you. So Cairns Regional Council over the last probably year has been looking at trying to understand what is going to be our best option for managing our organic waste into the future. And so we've started on a journey, I suppose, to map out those options. This is just a really short presentation just to let you know where we're at with things. If I can't get it to work. Oh, yep, there it goes. So I'm just going to very quickly mention what our drivers were to start this, our methodology, where they've got to so far in the consultancy, and some technology options, and importantly, the one that I'm most interested in is around, I suppose, future-proofing our decisions. Stop it. So importantly, from our perspective, our key driver is probably that our current contract for managing our organic waste stream from our curbside is due to expire in about 2026, which to some people might feel like a long time away, but when you're talking about major infrastructure investment, it's not that far away. So being a council, we're quite fortunate. We look after the waste management and the wastewater management. And so we're using this consultancy to map out all of our organic waste streams and to see where we can get synergies and you know, to see whether we can actually manage this waste together, or what are the risks of managing both biosolids and organic curbside waste together, and what are the costs and what are the legislative constraints, I suppose, around managing the different types of organic waste. So yeah, our key drivers were to make sure we're well set up for a future beyond our current contract for the management of our organic curbside waste, and also to try and improve our resource recovery efficiencies. So the process we've followed, because it's quite a unique project for council, and it's what we would call it's a strategic planning project, and it comes with a lot of complexity, both technically and in a policy context. We put quite a lot of effort up front into planning out what the project should look like, and I would probably argue we often don't do this component well for projects, and I think it's really important for these types of projects. So we've ended up with a project team that that has got a range of skill sets, from policy legislation type skill sets like my own to very technical skill sets around biosolids or curbside collection of organic waste. So there's many different components, and it's good to bring the different ways of thinking together to hopefully maximise or reduce the risk of making a bad decision in the long run. So once we worked out exactly what we wanted, we put together a request for quote, and we chose to put that out to, I think, probably 4 or 5 consultants that we knew had experience in this space. So they either had really good experience in the management of curbside waste or management of biosolids. Not many had good experience in both, but we put out a scoping document to consultants and sought obviously quotes based on that scope. So we chose a consultant that we thought would give us a really good outcome in terms of an Australian context, but also had an international, were able to bring a strong international experience into the decision making for us. So we've gone through now, we're actually just at the end of stage 2. So we're underway in the consultancy, where the first stage basically defined what the resource was, what did it look like from Cairns' perspective, and benchmarked some of the options that were available. Stage 2 is started to look at the feasibility of options and to start undertaking the market research. Stage 3, which we haven't got to yet, is where they will be doing an options assessment on the different options that we may have. Stage 4 will be the business case, and stage 5 is the recommendations report. This is just very quickly going through what that looks like. So the consultancy actually started in December, and like I said, we've just finished stage 2 of the consultancy. We have already started to run into a few problems in terms of our original scope, and that comes down to the complexity of the actual problem we're trying to solve. That when you put these, I suppose, these sorts of problems out to consultants to provide information on, if you haven't got the scope right, you can get some really big holes left in the analysis, which for a consultancy like this, we don't want any holes left in the analysis. At the end of the day, we want a high level of confidence at what will happen to all of the waste streams, regardless of what technology is chosen. So just quickly on the Cairns context, they went through and identified all our organic waste streams, and like I said, you know, there's green waste, there's the organic waste in the curbside collection, there's biosolids, there's grit and screening. So this slide just shows where we ended up with for Cairns. And I found this interesting. These are just some snapshots of different data that they've collected, and surprisingly, we have an advanced resource recovery type treatment process for our curbside waste collection in Cairns, and you can see that with the two bars at the left of the slide, you can see that we really only get about 50% recovery. So also in terms of what goes to landfill and what is recovered, once again, it's around 50%. With biosolids, currently in Cairns, the majority of our biosolids go to broadacre farms. So we actually get very good resource recovery, but you know, there are risks around that, especially with the introduction of the end of waste code. And also some of our biosolids, the bit in yellow is where they use biosolids as a feedstock to our organic processing facility for our curbside waste. And the bit in red is where we do sometimes get batches of biosolids that are too contaminated, in particular with arsenic, to be able to be used on farms, so it goes to landfill. So we have to take all of these factors into account when we're looking at what is the best option for Cairns. We then, and this is where the consultants are at the moment, looking at what technological options are available, and I found this summary really, really quite interesting. So the different options at the top of the slide around composting and anaerobic digestion, they still get quite low recovery rates. So you can see 50 to 60, 60 to 70% diversion rates, whereas the waste to energy, the gasification, pyrolysis get much higher diversion rates. So if one of our drivers is diverting waste from landfill, this is the sort of data we have to take account of. So, you know, another one of our objectives is to get the greatest beneficial reuse from the waste. And some people would argue those gasification or, you know, waste to energy options are not necessarily the best way to get to reuse a resource, but you know, sometimes you have these competing factors. So in this stage of the consultancy, they're looking at what processing, collection and production technology is available and tested in Australia and overseas. You know, what collection systems are being used and what's relevant. Interestingly, the consultant that we're using has a lot more experience in cold weather climates, and so you know, we've had to make sure they understand the difference in curbside collection in the tropics versus the cold weather. And they've also started to look at potential markets in this region, and also whether other councils are interested in collaborating with Cairns on this options. Finally, they've started to develop a probably a range of about 8 to 12 options that will go through a more fine-tuned assessment process. We're yet to get to an agreement on what assessment criteria we will use, what type of scoring we will use, and how we will shortlist, but that is all coming in the next part of the consultancy. The part that I suppose I'm most interested in is trying to understand and reduce the risk of the different parts of the puzzle resulting in a poor decision being made by council, because in the end this is going to be a very significant infrastructure decision. It's probable that we will end up managing the curbside organic waste separately to the biosolids, so there will be infrastructure required around both waste streams, and we're still looking at, you know, decentralised versus centralised. So it's a big expense for council, and it's something that, you know, once we get the technology, it will be in place for a long time. But we're trying to make these decisions in a very fluid policy environment, like with the End of Waste Code and the Queensland energy from waste policy, and you know, there's so much change in that policy world at the moment. It's very hard to make robust decisions. There's still a significant gap in our knowledge in terms of I suppose what the different technologies might mean, especially for things like emerging contaminants. So most people probably know there is some, you know, small amount of research happening around the place on looking at what the different technologies will mean for emerging contaminants like PFAS and microplastics and many others. We are, I suppose, on this issue, probably more than many others, I feel there needs to be a really strong emphasis on collaboration, both with the universities and with other utilities. Research in this space is very expensive, and so, you know, none of us are going to be able to afford to go it alone. So in saying this, we're in the early stages of looking at whether we can do a collaboration with some of the North Queensland councils around understanding the impacts of gasification on emerging contaminants in this more tropical environment. And just to finish off, I suppose, to bring it back to COVID-19, one thing I've noticed in the last, you know, 3 or 4 weeks, and I've spoken to Rob about this, is that councils are, you know, very much focusing on essential services and looking at reduced budgets and that sort of thing. And already I get the feeling that the priority for doing research will be less. However, I feel, you know, we're in a time where we actually need to be probably doing more research and collaboration, and I think that's a great risk for us, because some of these issues take time to resolve. They're not simple, and you know, we won't be able to just have an answer tomorrow. If we reduce the amount of research we're doing, we're not going to have the answers that we need for making good decisions in the future. The other thing I think with the COVID-19 is there's obviously going to be how we manage our biosolids and reuse effluent, and that's probably going to be far more sensitive, even if it's a perception thing as opposed to fact in the community. The way we manage these resources is probably going to be more sensitive. So we have to try and pull all these factors into our roadmap to make sure at the end of the day the decisions that we're making are robust for the future, so future-proofing those decisions, because it's going to be very expensive technology. I think that's all I had. So thank you.
Thanks, Lynne. That was fantastic. So we'll just invite questions from the audience. I've got a couple to get us started though. So let's go with it. How important is the treatment level of biosolids in terms of water content in affecting the costs of your roadmap?
Sorry, can you say that again?
So how important is the treatment level of your biosolids regarding water content, and how much does that impact the costs of your roadmap?
I suspect we haven't got quite there, haven't got there yet, but I suspect it will be fairly significant. And I think, you know, from speaking to our the engineers in this, you know, that have more experience than me in this part of it, is that I foresee that we will probably need some sort of technology at each of our sites to reduce the water content, and then potentially have a centralised processing facility. That's my instinct of how things will go. So we will probably have to, without predicting the, you know, what the outcome, but I suspect we will be doing some infrastructure works at each of our sites to dry out the biosolids more before they go to some sort of centralised processing facility.
Yeah, excellent. And so I've got some offers from the audience of various utilities that are happy and keen to collaborate.
Oh, really?
So yeah, that's good news. They're rolling through. But also,
I mean,
just
sorry, can I just say, Ryan, it's brilliant. I think more than ever we need Queensland Water and WASA to be playing a really strong facilitation role in this space, because it is really hard, especially, you know, we're a long way from everyone, to be keeping up to date with things, and to, you know, we don't want to reinvent the wheel or be doing research that others are already doing and that sort of thing. So I really value that facilitation role that you and Rob and Dave have been taking over the last couple of years in this space.
thanks for that. So we've also got a poll to run, but I'll just run through some more questions. So do you think the public perceptions about biosolids waste is changing because of COVID?
Well, I don't know. I haven't heard anything, but I just suspect any of the general public that actually think about biosolids and land application, which probably aren't many. I suspect, my instinct is that there will be a bit more sensitivity around it. So it's obviously probably perceived sensitivity, but, you know, perceived is as good as fact. And I mean, already we're seeing a trend of, in the major supermarket chains, and that not wanting to take fruit and vegetable crops that have had biosolids applied to them and that sort of thing. So my instinct, once again, is that with our biosolids, we will be, and I might have to eat my words here, but I suspect we won't be going for a land application option, because the risks are too uncontrolled. What we know now, or what we don't know now, which we might need to know tomorrow around different types of contaminants, to me puts too high risk in that area.
Yeah, thanks for that. And for those listening, if you're finding similar, you know, potential community impacts within your councils, please let Rob and I know. That just gives us a better awareness of what's going on regarding the biosolids. So we've touched on emerging contaminants a couple of times throughout Lynne's great presentation. So we'll just run a bit of a poll and just getting a bit of a feel for what the audience, what their level of concern for contaminants in wastewater, biosolids and recycled water are. Unfortunately, Lynne, as a presenter, you can't participate, but that's okay.
Oh, I like participating in the poll.
Yeah. Yep.
Give you a chance later with Lynne.
Okay, thanks.
So I'll just run this for another 45 seconds there.
we're particularly interested in this because of the consortium for contaminants of emerging concern that's just starting up at the moment. Cairns is involved in that, Lynne's involved in that, along with Townsville and a few other regional councils, and Toowoomba and Redlands as well, Mareeba. We see contaminants of emerging concern as being an ongoing issue, and although COVID isn't really in that group, the situation that we've seen with misinformation and myths and breakdown in communication, and all of our members having to look up information themselves and probably waste a bit of time, whereas having it provided centrally would be useful. All those issues are kind of the same. So we're trying to learn from the current pandemic, I guess, and put those learnings into the new consortium.
Just on, I've had 2 people say they can't seem to vote. So usually you should just be able to click it in your browser as such. But we found that if your council's gone the route of very high security, it may not work as much. So if you're really ambitious with what's said, let us know. Otherwise, you can try being full screen with the poll and then clicking then. So just maximize that viewing screen and then try voting. Give it another, all right, give another 5 seconds for everyone. All right, and I'll just share those results. So we'll use this for future, to help us inform future talks. But you can see that obviously the emerging contaminants is still relatively high on people's agendas. So that's good indication for us and future talks.
Yeah, that's really good. Thank you very much. Yeah, interesting. It's not a key concern in some places, but obviously not too many. And whether it's due to COVID or not, a few people said it's not COVID in the comments as well, which is kind of what we expected. So thank you very much for that. We've only got 2 minutes left, so I might close it out. If you have any questions or suggestions or feedback, please feel free to share them either through the go-to process or just by contacting us directly. We've got a couple of really good talks again next week, same bat time, same bat channel. We've got one from Martina De Silva from WSA, who's talking about a technology for treatment of water in small locations, small towns. A point of entry drinking water system for rural areas using IoT is the official title. That's a really interesting technology, and we've been working to try and get a demonstration of it in Charleville. So if you're interested in affordable ways of treating small volumes of drinking water, join in for that. And we also have Anna Whelan from Townsville talking about the research that 2 PhD students are doing there, and Townsville's collaborating with and actually running, and a new project that's just been kicked off now, which Lynne hasn't mentioned. She's saving, keeping the powder dry for Anna, I take it, Lynne.
Yes.
And talking about contaminants and the work that's being done, particularly at the Cleveland Bay STP. So join us next week if you're interested in those topics, and thank you very much for your time today. Do you have any last closing comments, Ryan or Dave?
Just noting that we'll update the next week's registration link in the media invites, and it'll obviously be advertised within the e-flashes and Zoho forum.
I'm good. Well done.
Good luck with the isolation. Sorry, Dave. Talked over you.
No, I was just saying, well done. It was great. Thank you. I'm good.
All right. Thanks everyone. Stay safe and enjoy the waiting period till we see what happens next in these exciting times.
Thanks all. We'll end it there.